with respect to immune-mediated diseases. Next week or within 2 weeks, I think I will come back on that issue again. However, my research is focused on another issue, especially on immune regulation. And since this lecture should be within the scope of neurodegeneration, I will tell you about multiple sclerosis, which is one of these diseases. And I also will talk about possible therapeutical possibilities with vitamin D, just an environmental factor which is readily available and very cheap. So altogether I will talk about these three entities, vitamin D, multiple sclerosis and immune regulation. And since the academic center for multiple sclerosis is not within Maastricht, but it's in the Orbis Center in Sittard, this is also part of the collaboration. Okay, if we... This is not first. There is apparently no control for this. But anyway, if we talk about vitamin D, we should be aware that there are two main sources of vitamin D. One of them is within the diet. So fish is containing, especially fat fish, is containing a lot of vitamin D. And in the previous century, many young people had to eat cod oil before they went to sleep, especially in winter time, to have sufficient vitamin D levels. Nowadays you have this vitamin D readily available in pills, and it's much easier and not as nasty to take. However, the main source of vitamin D is sun exposure, and especially the UVB exposure. And as you know, of course, sunshine is especially present in summertime, at least in this area of the world. So it's season related. Especially in summer, you have high levels of vitamin D because you have high levels of UVB exposure. However, in wintertime, like now, you don't have a lot of sunshine, and if there is sunshine, the UV irradiation that reaches the planet is too low to generate vitamin D synthesis. So you can go into the sun, but it doesn't help. Unless you go into the mountains, not the Dutch mountains, but the higher mountains, of course. If you go skiing, then you have again a lot of UV irradiation and of course vitamin D synthesis. Now, when you synthesize vitamin D in the skin or when you eat a lot of fish, you first have to metabolize this component, this vitamin D. You have to hydroxylize it, and that happens in the liver. Then you have 25-hydroxy-D, but it's not only produced in the liver, it's also produced by macrophages and dendritic cells, two main cells of the immune system. But even then you are not ready. Still, this is not the active component that is doing the job. You do need a second hydroxylation, and that apparently occurs in the kidneys. So then you have one 25-hydroxylated vitamin D. And this is actually the active component, but now, as you can see, it's predominantly metabolized in the kidneys, but also all cells of the immune system, especially also the lymphocytes, can do the job. They have the enzymes, the machinery to do this. 1,25-dehydroxyvitamin D, as I said, is the active component, and that can bind to the vitamin D receptor. The vitamin D receptor, however, is not a regular receptor on the surface of the cell, but it's an intracellular protein. So the vitamin D first has to go into the cell and then can bind to the receptor, and then this receptor will function as a kind of transcription factor and regulate multiple genes. More than 100 genes contain so-called vitamin D response elements, and therefore the gene expression will be upregulated or downregulated. We know the vitamin D metabolism to be very important in calcium metabolism, so in your bone health, but it appears that vitamin D is also important in cell proliferation, and as I will discuss today, it's very important in immune regulation. And for that, of course, you have to understand that these cells indeed can take care of their own production in their local environment. Now, Now, coming to multiple sclerosis. Multiple sclerosis is a severe chronic disease. It's a demyelinating disease of the central nervous system. And if you look on MRI, you see multiple white plaques. And that is, so multiple sclerotic plaques where there is tissue injury, and especially the myelin sheets along the nerves is destroyed. This disease onset is typically between 20 and 40 years, but you may also have disease onset during childhood or even further at older age. In the Netherlands, we have about 16,000 patients, so that means that 1 in 1,000 is having this disease. This is not the yearly incidence, but it's the overall prevalence of the disease. We think it's having an autoimmune origin. It's definitely true that multiple sclerosis is mediated by the immune system, but for the origin we are not quite sure if it's autoimmune or other types. But we know that in the disease process, T cells are very important. I will come back to that later. With respect to the etiology, as I mentioned, there is not a lot known. We know that genes are involved, as in many diseases. If you look for twins, you see that there is a higher chance that, especially in case of identical twins, that the brother or sister has also the disease. But there is more than just genes. Within the genes, there is one strong gene association with HLA-DR15. And DR15 is an MHC class 2 molecule, and I think you have had already the discussion on MHC. So MHC class 2 interacts with CD4 T helper cells. So the strong interaction with this DR15 tells us that indeed T helper cells are also very important in the disease. But in the environment, there is also a lot of evidence that vitamin D is associated with the disease. But besides vitamin D, we also know that, for instance, Epstein-Barr virus infection is associated. So there are a lot of studies there as well. Smoking is associated, so there are many more things than just vitamin D. If we talk about MS, you have to realize that the disease can present in two different ways. It can present as a kind of relapsing remitting disease, relapsing remitting MS, and in that case you have disease exacerbations like here, and then there is full recovery. And then you may have an exacerbation again, full recovery, etc., etc. However, on the long term, the disease will become progressive, and that means that there is no restoration to normal again, but you see that there is increased injury in the brain. And eventually, there is even not a distinction anymore between relapses and remissions. So, and in this stage, we talk about secondary progressive disease, because here there is no relapse anymore. It's called secondary progressive because there is also another form of MS which is called primary progressive, which means that this system already starts from the beginning. So these patients do not have relapses, remissions, but they have steadily increased in demyelination in the brain. Next to that, we know that especially in the relapsing remitting form of MS, the first stages are inflammatory stages. So in the first years of the disease, it's a matter of a lot of inflammation which is going on, and later on the inflammation goes down, but you have increased amount of demyelination and therefore scar induction and less functionality. Now, if we look for the immune system, how is it involved? We know that the trigger for MS does not take care within the central nervous system, but it happens in peripheral lymphoid tissue, especially the lymph nodes. There, T helper cells are activated or primed by antigens, and we don't know whether that are antigens that have leaked away from the central nervous system, or for instance are antigens coming from Epstein-Barr virus, but it's evident that at least here the first priming takes care. And if you have T cell priming, T helper cell priming, there will be skewing to certain T helper subsets. And I hope you have had already the concept of Th1, Th2. Is that right? Okay. So primarily we thought that MS was a typical Th1 mediated disease, where Th1 cells produce a lot of interferon gamma, and they also stimulate the cell-mediated immunity. However, a couple of years ago it appeared that it's not just Th1, but it's another subset which is referred to as Th17. Th17 will not mean that in the meantime we have generated 17 different T helper subsets, but we stopped at Th3. So we have Th1 for the cellular immunity, Th2 for the humoral immunity, and Th3 was coined to have a kind of regulatory function. But Th17 was called Th17 because it produces a cytokine IL-17, interleukin 17. So that's the only reason. But this seems to be a more pathogenic cell than the Th1 cell, at least within MS, but also in other autoimmune diseases. This has appeared to be a dangerous cell type. Anyway, we also thought for a very long time that the central nervous system was a so-called immune privileged site, meaning that the immune system could not enter the central nervous system, and that could make sense because if you have strong inflammation within the central nervous system, that would clearly affect the function, and therefore it was not compatible with life. However, it appeared that there still is some low-grade immune surveillance by memory cells also within the central nervous system. So those cells that have been primed and have been differentiated into memory cells are able to go into the bloodstream and pass the blood-brain barrier and enter the central nervous tissue. If doing so, they may encounter their antigen, the myelin-associated antigens, and if that happens, they become activated and they start to produce all kinds of nasty cytokines like IL-17, and the whole immune system will be triggered to destroy the central nervous system. So then it's not only T cells, but it's also a lot of macrophages that enter the brain and are involved, but also B cells are involved. There is increasing evidence that besides T cells, also B cells play an important role in multiple sclerosis. And as said, eventually the myelin sheaths along the axons will be destroyed. And that prevents or that strongly delays the signal transduction from the cell to the end of the axon. Now, the outcome of that is that if you don't have appropriate signal transduction along the nerves, that there will be weakness of muscles, and that's what you experience. So you have motor weakness, so that's muscle weakness in about half of the patients, but other patients have sensory problems. Also, there's very common that people have problems with the optic nerve, so they have blurred vision, and several other things that are associated with this effect in the central nervous system. Now, coming to the relation between vitamin D and multiple sclerosis, this is a graph which shows the world, of course, and as the distribution tells us that as closer you are to the equator, the less chance that you have to develop MS. So especially in our countries, you have a relatively high chance to have MS. Now obviously you can say that people in Europe are genetically completely different from people in Africa. So from there, you don't know exactly whether it's a geographical difference or a genetic difference. However, there is one big advantage. There is one huge country, which is called Australia. And Australia more or less has the complete range of being close to the equator and far away. Especially Tasmania, one of the southern islands, has an MS incidence very close to what we have in the Netherlands. Now, why is this an advantage? Because nearly all people in Australia are founded by people from Great Britain. So they are genetically quite homogeneous. So, and from there, therefore, we can postulate that it is indeed a geographical difference that is associated with MS and not a genetic difference. Now, if we look for sun exposure and relation with disease severity, you can look for exacerbations, how many relapses are experienced by the patients. You can look for cytokines, especially inflammatory cytokines, and you can look for MRI lesions, so the white matter lesions you see typically on MRI. And if you look at that, you see again, in this case, it's on this axis the vitamin D, on this axis the MRI lesions, and you see there is a very close association. So if you, and here you have to be careful with interpreting this graph, the y-axis for vitamin D is inversed. So here you have low amount of vitamin D, here you have high amounts of vitamin D. So it appears that if you have low amounts of vitamin D, in January, February, you have increased lesion numbers based on MRI. And you can see the same phenomenon with inflammatory cytokines and exacerbations. While in late summer, early spring, when your vitamin D levels are highest, at least on the northern hemisphere, you can see that there is a clear decrease in the number of lesions in the brain. So again, here you can see an association with vitamin D levels and disease severity. This goes on because even if you look at the date or the month of birth of a person, it appears that patients that have MS are more likely to have been born in late winter, early spring. So when your vitamin D levels are low. So apparently very early in life you already do something with your immune system. You see similar things with asthma and allergy that makes you prone to develop an autoimmune disease. And this goes on. If we talk about vitamin D and risk of development of MS, there is, as I showed you, this north-south gradient and MS prevalence. Especially, of course, this is in the northern hemisphere. In the southern hemisphere, it's just the other way around. There is a negative correlation between UV levels in a region and MS prevalence. So it's not only north-south, but it's also associated with UV levels. And it appears that not only with the month that you are born, but also sun exposure during childhood protects from MS. And this is shown in this slide. So the ones that have, this is here. The lower adolescent vitamin D levels are more prone to develop MS later in life. So the ones that have very high levels of vitamin D during adolescence, they have less chance, reduced chance to develop MS in the end. This is not absolute. As I said, there are many more components that are involved in MS development, but at least it shows very clear-cut associations between vitamin D and MS. It tells us also something about exacerbation, so the relapses. In a study that we did in Maastricht, we found that, and that was a rather small study, that vitamin D levels predicted more or less the amount of relapses that a patient had. We did it the other way around because it was a retrospective study, and what we showed is that the patients that were relapse-free for at least 2 years had much higher vitamin D levels at the start of the relapse measurements. However, this only was true when patients were included had a very short period of MS, so only less than 5 years. If you included all patients, you didn't see this association. But this tells us again that MS is an inflammatory disease during the first years, while later on it's a more demyelinating disease, so completely different. So apparently vitamin D seems to be especially efficient in protecting when you have short duration of disease. Now later on in Tasmania, now remember Tasmania has more or less the same disease incidence as what we have in the Netherlands, they did a longitudinal study with a lot more patients. They followed up the patients for 2.3 years. And what they showed is shown in this figure, is that with increasing vitamin D levels, you have a lower hazard ratio of having a relapse. And it tells us that every increase of 10 nanomole per liter, you have a 10% decrease of chance of having a relapse. So that would tell us, if it was indeed a linear relationship, that if you could increase your vitamin D level with 100, you almost completely can prevent relapses. But of course, then you assume that this indeed is a linear correlation. Typically, in a United States study, they found exactly the same. So also with every 10 nanomolar increase, they have about 10% reduced risk of having a relapse. So even in other areas, they could confirm these data. Now, how does it work? It's an hypothesis. If you have an immune response, T cells need to encounter an antigen-presenting cells, and they have to have an interaction with the T cell receptor and the MHC with the peptide in it. That will result in activation of this T cell. And actually, if a T cell becomes activated, it starts also to upregulate the machinery to metabolize 25-hydroxyvitamin D into the active component. And if that happens, so both cells can do the job, they also both have the vitamin D receptor, and if that happens, the lymphocyte will respond by reduced proliferation, reduced amounts of pro-inflammatory cytokines, reduced antibody production, increased anti-inflammatory cytokines, and also a more regulatory phenotype. And also the same holds for dendritic cells. So basically what we state is that what we state is that there is a kind of negative feedback mechanism on immune activation by vitamin D. And the cells of the immune system take care of this negative feedback mechanism, and therefore you have tolerance or at least homeostasis within your immune system. However, if you lack 25-hydroxyvitamin D in your circulation unit in your body, so if you don't have enough sun exposure or fish in your diet, then you don't have these components, and then there is no possibility to generate this active component that can take care of all these functions. And then you don't have homeostasis in your immune system, but you may have autoimmune diseases. And as mentioned, it may not only be autoimmune diseases, it may also be allergy and asthma and other kind of nasty things where your immune system goes in the wrong direction. Don't try to read this slide. This is just a slide where they did a genetic risk analysis. And for us it was a very interesting paper. It was published about 2 years ago, I think, where they included many thousands of patients with MS, something like 20,000. They did a genetic profiling. They also included 10 or 20,000 healthy controls, and they tried to find out which kind of genes were involved in the process of multiple sclerosis. Now, there came a couple of interesting things out of this study. First of all, they identified about 30 genes that were associated with MS. One of them I already mentioned, which was HLA-DR, and HLA-DR15, it has a very, very strong association, much stronger than all the other genes together. The other genes only have a very minor association, and they pop up just because they have such a huge amount of patients and controls. But nevertheless, it appears that those genes that are associated with multiple sclerosis, 1/3 of them are immune system genes, affected by cytokines, co-stimulation, whatsoever. And the other thing which was interesting for us, there are two genes. I don't expect you to read it, but these are the two enzymes that metabolize vitamin D. So the one that produces 25-hydroxyvitamin D and the one that produces 1,25-hydroxyvitamin D. So this again illustrates that there is a clear link with vitamin D and MS and the immune system. Now, before we go to humans, we have to go to animals. And here they did an experiment, and the animal model, I have to say, for multiple sclerosis is so-called EAE, which stands for experimental. And here I hesitate what to say, because in all the days, the original term was allergic encephalomyelitis, because originally one thought that MS was an allergic disease. But nowadays, this EAE is referred to as experimental autoimmune encephalomyelitis. Nevertheless, in this case, there is a mouse, and this mouse is transgenic for a T-cell receptor that recognizes a typical antigen for MS. So all T-cells in this mouse are specific for this T-cell receptor and therefore are specific for this peptide. Is MS-inducing peptide. So if you then immunize the mouse with that specific peptide, all T cells become activated and will try to go into the central nervous system and destroy the myelin sheaths of the nerves. And what you will see is that you have this MS-like disease. So the open circles tell you that there is disease onset after about 10 days. Then there is a kind of recovery, but you have relapses, recovery, etc., etc. So in that sense, it really resembles MS. Also, if you go and look in the brain, you see some similar pathology. But here I want to tell you that if we treat these mice with the active component of vitamin D, so the 1,25-dihydroxyvitamin D, starting from day 0, so starting from the time that we do the immunization, there is no disease whatsoever. There is a minor inflammation going on, but you really can prevent disease development just by giving the active component of vitamin D. Now, if that is true, we also can try to learn what the mechanism is of this vitamin D. So, additional experiments were done in the same system, and in this case, it's the same story as what I showed you on the previous slide. So I do have a mouse which is transgenic for the T-cell receptor specific for an MS antigen. If you give active component of vitamin D, you can prevent EAE. If you don't give it, the disease will develop. However, if I now take this mouse, but make it deficient for RAC2, and this stands for recombinase activating genes. Anybody knows what these genes do? They are very important in generating T-cell receptor and B-cell receptor genes. And if a T-cell or a B-cell during development is not able to produce an adequate T-cell receptor or B-cell receptor, it will not survive in the thymus or the bone marrow. So the consequence is that if I have a RAC knockout mouse, there will be no T cells or B cells whatsoever in this animal. So now I start to immunize again, and what I see that even if I give vitamin D or the active component, there is still development of disease. So the conclusion from this experiment is, I forgot one thing I have to say, because this animal was infused with T cells that were MBP specific, because it's transgenic for this one. So this animal does have T cells specific for the antigen, so they of course are needed to cause disease, because they are the pathogenic cells, but there are no other B or T cells because the animal is RAC deficient. And apparently, this active component of vitamin D acts on the other B or T cells to prevent disease development. And if they are not there, you cannot prevent disease. Similarly, and this is a somewhat different system, but again, it's another model of EAE, but now they induce disease with another myelin-associated glycoprotein. Essentially, it doesn't matter. Again, of course, you can see that if you give a placebo, the animals will develop disease, as you see here, while if you give the active component of vitamin D, there is hardly any disease whatsoever. Now this animal, they used for another experiment, that is, that they used it in a setting where they made the animal IL-10 deficient. So this animal is exactly the same as the previous animal, but now it cannot produce IL-10. And IL-10 is considered to be a cytokine, an anti-inflammatory cytokine, which is very important in immune regulation. Now, as you can see, if the animal is not able to produce IL-10, I can give the active component of vitamin D, but the animals will have disease in the end. In a similar way as the placebo group. And also, if I make an IL-10 receptor knockout, I have the same phenomenon. So from these two experiments, I can conclude that besides lymphocytes, also IL-10 is a very important component in the mechanism of vitamin D effects. And since in the 90s of the previous century and early in the first years of this century, there was a huge bias in research. Everybody thought that everything in the immune system was taken care of by regulatory T cells. Did you discuss already regulatory T cells? Okay, so very shortly, it's a cell that suppresses immunity. Nothing else. However, in the 80s of the previous century, we thought there was a suppressor T cell, which was within the CD8 T cell compartment. But since nobody could prove the identity of this cell, the term suppressor T cell was banned. It was not allowed to use it anymore in the literature. So when this cell showed up, which is a CD4 T cell, it's not a CD8 T cell, we couldn't refer to it as a suppressor T cell, although we should realize that regulation can go in two ways, activation or suppression, but the only thing that this regulatory T cell does is suppression. So basically it's a suppressor T cell. So the idea is indeed that vitamin D affects the regulatory T cells in MS and in many other diseases. And the concept I always try to use to explain what these regulatory T cells do is, if you have the concept of Th1 and Th2 on a balance, we know that Th1 cells can give cell-mediated immune responses. Th2 responses are humoral immune responses. And what these regulatory T cells do is take control that these two responses are not exaggerated. And you can see that in the next slide. If the control of regulatory T cells is too little because you have too few of these cells or they don't function as good as they should do, you may have exaggerated Th1 responses, meaning autoimmune diseases, transplantation rejection, or you may have exaggerated Th2 responses, like allergy, asthma. On the other hand, if you have too many of these regulatory T cells, or they function very efficiently, your immune responses are very minor. And this may be insufficient to get rid of an infection. It may also be insufficient to have a good response on vaccination, and it may also be insufficient to have a good immune surveillance for tumors. So altogether, you should have a balance between Th1 and Th2, but you also should have the appropriate amount of function of these regulatory T cells to have clear homeostasis in your immune system. And therefore, it is often suggested, of course, that in autoimmune diseases, the cells are present in too little amounts or they don't function very well. Now, many studies have been performed on these regulatory cells in multiple sclerosis. As mentioned, it's just a suppressor CD4 T cell. The studies have been enabled by finding markers how you can identify these cells. One of these markers is IL-2 receptor, which is CD25. This was a very cumbersome marker because in the mouse it worked very well, but in the human, if T cells become activated, they also start to express IL-2 receptor. So then, in activated circumstances, you cannot distinguish anymore between regulatory T cells and activated T cells. However, 10 years ago, there was discovery of this marker, FOXP3, and FOXP3 is a transcription factor which is very specific for these regulatory T cells. It happens that there is an immunodeficiency which shows that patients that are deficient for this gene develop very severe pathology in the first year of life. They develop very strong diabetes, but they also have all kinds of allergic symptoms, inflammatory symptoms, etc., etc. And if you don't give them a bone marrow transplantation, they will die within 1 year. So this told us, and these children do not have any regulatory T cells whatsoever. So this is a very good marker. And more recently, also the absence of IL-7 receptor alpha is a good marker. So in this case, nowadays we often use the combination of CD25 and CD127 because they are both surface markers. And you can see that those cells that do not express CD127, this axis, and have a high expression of CD25 on the x-axis, these are the regulatory T cells. And within CD4 T cells, in the peripheral blood, it's about 5% that have this phenotype. And if you check these cells for FOXP3, they nearly all have this FOXP3 marker. I come back to that later. Now, it has been published that the function of these cells is reduced within MS patients, although the numbers are normal. They also have a reduced function during relapses, and it occurs again, especially in patients with a short course of disease. So again, within the phase that you have this inflammatory disease. Now, how do you study the function of these regulatory T cells? That you can do that in vitro by mixing cells. And what you do is you purify responder cells, and as responder cells we talk about CD4 T cells that at least do not express CD25. And for the regulatory T cells, we define them as having CD25 high and no expression of CD127. So the one that I showed you in the plot in the previous slides. And then you can mix them in certain amounts, in certain combinations, and you can look whether the regulatory T cells are able to suppress T cell proliferation of the responder cells, because the responder cells are activated by an antibody against CD3. So now, in this case, you only have responder cells. So and if you label the responder T cells with a dye, and every time that the cell, so if the cell gets activated, it will start to proliferate, and every time that the cell will proliferate, will divide, the dye will be distributed to the both daughter cells. So it will be half the amount. If they proliferate again, it will be again half the amount, etc., etc. So therefore, if there is strong proliferation of the cells, you see a gradual decrease in the amount of fluorescence. So these cells have divided once, these twice, 3 times, 4 times, 5 times. And this is some trash which was from the feeder cells. Now you start to add in increasing amounts of regulatory cells, and that's what you see here. So you see an increasing amount of unlabeled cells. These are the regulatory cells. And if you see on the other side of the spectrum, if you have twice as many regulatory T cells as compared to responder cells, there is no proliferation whatsoever. If you have less cells, you still have some proliferation. Be aware, in this situation, you still see some effect, but here I have, for each regulatory cell, I have 4 responder cells. So that means that within the CD4 compartment, about 25% are regulatory T cells, while in real life, it's only about 5%, so it's even 5 times less. So it's quite tricky to have small effects on these regulatory T cells to manipulate T cell responses. It's never as extreme as what we see here. This is what happens in vitro. Now, these experiments we also used in 30 MS patients. So we had increasing ratios regulatory responders, and we looked for the percentage suppression. So if you have only few regulatory cells, you have a little suppression. If you have a lot of regulatory cells, you have a lot of suppression. From this, we calculated for each patient the amount of regulatory T cells you need for 50% suppression. And that's what is shown here. So this is the regulatory responder ratio for 50% suppression. So the ones that are on top here need a lot of regulatory T cells to have 50% suppression. So the regulatory T cells from these patients perform rather bad, while the regulatory T cells from these patients perform very well. As mentioned, they are all from MS patients. Then these values we correlated with the vitamin D levels in the patients. And what you can see, there is a very nice correlation that those patients that have a bad regulatory function have low amounts of vitamin D, while the ones that have a good regulatory function have high amounts of vitamin D. So apparently, indeed, the function of the regulatory T cells appears to be associated with the amount of vitamin D in the circulation. In this case, we have drawn a linear relationship, but of course it may be different as well. It may be like this. We don't know. We need many more points for that. But this was very promising in order to do a bigger study. But besides that, we also looked at the cytokine expression. As I mentioned, interferon gamma, typical Th1 cytokine, IL-4, a typical Th2 cytokine. You can also look for the Th1-Th2 ratio, so the interferon gamma IL-4 ratio. And again, you see that this ratio is high, so a lot of Th1 cells if you have little amount of vitamin D, and it's high if you have high amounts of it. It's low if you have high amounts of vitamin D. We also looked at the two other cytokines that are important in MS. We looked for IL-10, which is the anti-inflammatory cytokine. We didn't find any association there. And we looked for IL-17, because IL-17 is nowadays considered to be the most pathogenic cytokine, representing the so-called Th17 alpha cells. And interesting, for Th17, there are studies that say that if you look within the regulatory T cells, so again here we have this marker CD127 versus CD25. CD25 high, CD127 negative cells are considered to be the regulatory T cells. If you gate on these cells and you look for expression of CD39, I will now explain what this marker means, but you can see that there is a population clearly negative and there is a population clearly positive. An experimental study showed that especially the CD39 positive regulatory T cells, but not the negative ones, are able to inhibit IL-17 production. So for MS, these CD39 positive regulatory T cells appear more important in suppression than the overall composition of the regulatory T cells. Now, for that reason, we looked for CD39 positive T reg cells within healthy controls, within MS patients in remission, or MS patients during a relapse, and as you can see, there is no difference whatsoever. Even if you express it as the number of the percentage of cells within the Treg compartment, there is not much difference, although it's typical that during a relapse, a subgroup having normal levels and this different group has very low levels, which even appears to remain low if this patient goes into a remission again. So these patients were measured twice, during a relapse and during the subsequent remission. And as you can see, the ones that were high in the CD39 positive Tregs remained high, while the ones that were low remained low. What it means, we don't know. Anyway, none of these was correlated with vitamin D. However, what we did see is if we look for IL-17 positive cells within these cohorts, you can see that especially both the patients in remission but also within relapse had many more patients with increased amounts of Th17 cells. So if you take this dotted line as a kind of cutoff, you see that within the controls there is only one, while there are multiple within the MS patients. Now, interestingly, if you look for the patients that are in remission, so that have no active disease, there appears to be a correlation between the percentage of IL-17 cells within the CD4 compartment and the CD39 positive Tregs. So the more IL-17 cells you have, the more CD39 Tregs you have. This seems a little bit strange because I told you that especially these CD39 Tregs are considered to be the ones that inhibit IL-17 producing T helper cells. However, we also noticed that this correlation cannot be observed during a relapse. So what we hypothesized is that during a remission, there is again a new kind of homeostasis in the immune system where you have balance between the CD39 controlling cells and the IL-17 pathogenic cells. So apparently it's a kind of feedback mechanism that you induce more regulatory T cells of this phenotype when you have a lot of IL-17 cells. While this control, this new balance in the immune system is lost again if you experience a relapse. And then you don't see anything of this. So knowing all this, we decided to start an intervention study and said, okay, so far we only found an association between vitamin D levels and the function of the immune system as well as disease parameters in MS. So what we wanted to do is we wanted to give patients with a short duration of MS, less than 5 years, again because that's the inflammatory stage of the disease, to give them with high amounts of vitamin D in order to improve the regulatory T cell function. So we took the risk actually to say, okay, we give very high amounts, which is 20,000 international units per day. And normally the current advice of the National Board for taking supplements is about 500 to 1,000 units a day. So this is very much more than just that. We did that for 3 months. And of course, because of the high amounts, we had to take care of safety. And we monitored these patients very carefully with respect to, especially to calcium metabolism, because that may be dangerous. And of course, we measured all kinds of immune parameters. As you can see, if you do this and you measure 25 hydroxyvitamin D, overall level starts at about 50, and you increase to nearly 300. So these are really high amounts of vitamin D in the circulation. Importantly, if you look for the levels of 125 hydroxyvitamin D, you don't see such a strong effect. It's rather stable during 3 months of time, indicating that there is very accurate regulation of this component, of the active component of vitamin D. And therefore, it appears that this strategy is also rather safe. The experiments that I showed you in mice, these were treated with the active component. If you do that in humans, you have a much bigger problem. But if you treat them with the precursor, it's more safe. And that it is safe, you can see in this slide, because if you look for calcium metabolism, the serum total calcium should not increase above this level. And it didn't, it was rather stable. Next to that you can measure the calcium level and the creatinine level, and you can calculate the ratio between these two. And the ratio should be below 1. So for the ratio you should look at the y-axis on the right side. And this is at week 12, so after 3 months of high-dose vitamin D treatment. None of the patients had levels above 1. We also followed the patients for another 3 months for safety reasons, and again, none of them had levels above 1. So even these very high doses were very safe, and in the meantime, some other studies have been published that confirm what we found. But more important, what happened with the immune system? I have to say that we, for logistical reasons, we only were able to include at that time 15, so 15 patients. We did again the same proliferation inhibition assay by mixing cells. And what you can see is basically that, you can look at this slide, we did not find a correlation between the Treg function and at least there was no improvement of Treg function 12 weeks after the treatments. Some patients increased, and sorry, I put it in the wrong way. What is shown here is the ED40, more or less the same as ED50, regulatory responder cells. Meaning that if the value is high, the function is bad. So you see that the majority of patients do show improvement of Treg functions, but some of them do not, even have worse function. If you look, however, which patients respond, that are the patients that are very low in vitamin D levels at the beginning of the experiment. So apparently you may have an effect in the ones that have a poor vitamin D status, but you don't have an effect in patients that have a normal vitamin D status. What happens with the Th1-Th2 ratio? You can measure again the cytokines, interferon gamma, IL-4, and from there you can calculate the ratio. Here we did see a decrease in the ratio, which was significant. So apparently you have less Th1 and more Th2, so a beneficial effect of vitamin D. And we also had a beneficial effect if we look for the IL-17 pathogenic cytokine, IL-10 ratio, which is the anti-inflammatory cytokine. I skip this one. So what I showed you is that from animal experiments we learned that lymphocytes are important and that IL-10 is important. And because of the time at that moment, everybody thought, okay, it should be these regulatory T cells. But you also have B lymphocytes that produce IL-10. And these are in focus more or less the last 3 years, and these are called so-called regulatory B cells, to make it complicated. And these regulatory B cells produce IL-10. So the conclusion from the animal experiments that it should be about regulatory T cells is not correct. It might also be about regulatory B cells. So why is this thought important? Because if you take mice that are completely deficient in B cells and you induce disease by immunization, they have far more severe disease than when they don't have B cells. So apparently the B cells can downregulate, can suppress disease manifestations. Additionally, if you transfer B cells that are able to produce IL-10 as well as TGF-beta, you can ameliorate disease, and this is dependent on certain kind of co-stimulatory molecules, but that's not that important. So again, you can see that B cells can suppress disease and that IL-10 may play a role there. So for that reason, we also looked within the MS population for these regulatory B cells, for IL-10 producing B cells, which is here on the y-axis. If you look in healthy controls, you can see that about 3 to 4% of the B cells have this phenotype, this function. While if you look in MS, whether it's in remission or in relapse, it's strongly reduced to less than 1%. So here we don't study the function, but we study the phenotype in terms of IL-10 production. Interestingly, if then you look for these B cells, if they are naive B cells or memory B cells, and you can look for naive memory ratio, it appears that the naive memory ratio of these B cells in the remission cohort is similar as in the healthy control cohort, while in the relapse cohort it's predominantly consisting out of memory B cells. So apparently there is not just a drop in the amount, but during relapse, during active disease, you also have a shift in naive versus memory ratio. And nowadays there are a lot of studies that are looking for these cells, but again we did not find a correlation with vitamin D. So, coming to the end of the story, I'd like to conclude that, and I hope I have convinced you, that there is a very strong association between MS and vitamin D status. I showed that there is a correlation between regulatory T cells, Th1, Th2 ratio, and the vitamin D status, at least in the physiological range. The number of Tregs as defined by the CD39, so the ones that are able to suppress Th17 cells, are not associated with vitamin D, but seem to have a kind of feedback mechanism in order to control these pathogenic cells. So if there are a lot of the pathogenic cells, you also have a lot of these cells. If you give a high dose vitamin D supplementation, you have a better homeostasis within your peripheral T cell compartment. We were not able to show it for the function of the Treg, but we could show it for IL-10 as well as Th1, Th2 ratio. This one I didn't show you. And as the last one, the number of Tregs are not associated with vitamin D, but they are definitely reduced within MS, and during a relapse they are shifted to a more memory-like phenotype. Now, and the eventual proof to show that vitamin D is indeed doing something with MS is by doing a huge study which is placebo-controlled and double-blinded, and that is what is currently in progress. So the idea is to include about 350 patients, and they all will have a high dose vitamin D. We didn't go up to 20,000, but in this case 14,000 units a day. We will follow this for 2 years, and hopefully we will see a reduction in the number of relapses, but also the lesions on MRI, etc., etc. Within a subgroup, we also will further study all kinds of immune parameters to have a better idea what this is really doing. And for the one person that is old enough in this room, in the 60s of the previous century, there was this guy, which was called Joris Liepinte, and he told us that you should drink every day 3 glasses of milk, and then you are going to be strong. Basically, this was a political advertisement because there was too much milk in Europe, so they wanted to get rid of it. But be aware, he was probably right, because also in milk there is some vitamin D, so we should have sufficient suppletion with vitamin D to remain healthy. Yeah. And to finish, these are all the people that were involved in our studies. I don't go through all of them, but I thank you for your attention.